If he passes.
If he passes.
If he passes, we're going to take it back. And we're expecting very high things because he also won the student research competition. So it better be really, and by the way, just walked in the other co-biology student. So Austin Cusick will be presenting today. Austin is a pre-med major. And Austin will be presenting the potential for flower nectar to allow mosquito-to-mosquito transmission of Francisella tularensis. Austin.
Hey everyone, thanks for coming. I know it's finals week or dead week, but I really appreciate taking the time out here to listen to this because I'm excited about it and I hope you guys are too. So this is the potential for flower nectar to allow mosquito-to-mosquito transmission of Francisella tularensis. Now, the first picture I want to show you, a little gruesome. This picture is of a child who was bit by a mosquito, developed this infection 7 days after, which then erupted into ulceroglandular tularemia, and even after antibiotic treatment, shown in this graph here, or this image here, still has adenopathy in the glands. So I just want you to, honestly, I want you to visualize, the mosquito transmission of Francisella tularensis, because this is dangerous, and this is an Eastern German boy, so this isn't really characterized in Eastern Germany. That's why this is prevalent, because this case is one of the first of its kind in that area. So first I want to talk about my outline, and I will go over a little background of Francisella, the mosquito, the flower nectar, and why, and answer these research questions that you'll see sporadically throughout the PowerPoint. Now the background. Francisella tularensis causes the disease tularemia, and tularemia is a highly infectious disease, so less than 10 bacteria are necessary to cause lethal infection, and some sources even say 1 bacterium is all that's necessary to cause an infection. So much so that it's categorized as a Category A bioterrorism agent by the CDC for its lethality and its use in bioterrorism, shown in World War II. Additionally, it can be spread many different ways. This can range from inhalation to mosquito bites to asymptomatic symptoms. Here are an example of some of those routes of transmission. We have oropharyngeal, which is eating contaminated meat. So typically mammals, like the rabbit that I have cutely pictured here, they become infected with Francisella tularensis, and then hunters that eat the rabbit and don't cook it properly, eat the meat, and then get an infection in their esophagus and GI tract. Human form is typically when Francisella tularensis is sprayed into the air and you inhale it, and then it colonizes your lungs, causes pneumonia. There's ocular glandular form. So ocular glandular form is an infection of Francisella tularensis just localized to the eye. Glandular form is a form of tularemia that just shows glands. There's no ulcer that's present. So you just have a lot of swelling in the glands that requires drainage and antibiotics. There's typhoidal, which is asymptomatic besides the fever, the sweats, the chills, and you don't even have swelling in the glands. And lastly, ulceroglandular, which was pictured with the small child prior. Ulceroglandular is spread most commonly by ticks in the United States, and the least appreciated vector is the mosquito. So mosquitoes have been now labeled to spread Francisella tularensis from host to host in the Scandinavian regions, Western Russia, and now Germany. So they're able to spread it, and we're trying to figure out why, because they're linked to many different outbreaks. And their role in this isn't quite well known. We don't know the different things about it. So for example, we don't know if it's a mechanical vector, which means the bacterium resides on the external surface of the mosquito and just in a sense hitches a ride. Or we don't know if it's a biological vector. We don't know if Francisella tularensis invades the mosquito, uses the mosquito to replicate rapidly, and then moves to different parts of the mosquito for further transmission. And that's something we need to delineate to properly target this disease. Now here's an example by Theolis et al. Theolis et al. Showed transstadial transmission of the bacterium across different life stages of the mosquito. So in nature, a larvae, that typically 80 species of mosquito larvae, would typically eat protozoans or biofilms in a pond, become colonized with Francisella tularensis, and have the ability to carry it through these different life stages here. And then infect, supposedly infect humans or other mammals. So Theolis et al., after showing this in nature through 16S ribosomal conformation of Francisella tularensis, they wanted to repeat this in a laboratory setting. So they co-cultured Aedes aegypti larvae with Francisella tularensis, watched it, and were able to recover Francisella tularensis in these different life stages over time. And not only that, they took it one step further. They let some adults feed on the blood meal and were able to show transfer of Francisella tularensis to the blood meal. So this shows incidents that it could potentially be biological. To make it through these different life stages, it cannot be on the external surface because the mosquito leaves, the shell behind in the water. And now the nectar reservoir. So we're going to add this new component to it, this new exciting component to it, and that's the nectar reservoir. So mosquitoes, if you didn't know, feed primarily on nectar if they're male, and intermittently feed on nectar if they're female. So this feeding on nectar, like transiently, can this allow for an exchange of bacteria? Additionally, can the nectar provide a conducive environment for bacterial survival? So think about nectar composition. Primarily simple sugars with amino acids and protein, right? So could these simple sugars and amino acids and protein be utilized by Francisella tularensis to survive for a longer period of time and even allow better transmission of itself? Now, I said a lot of words, and I get that, so I'm going to put it down in pictures. And no other way to do it but keep clip art pictures. So this top row is going to be the normal paradigm of mosquito transmission of a disease. You have an infected rabbit that appears, and a mosquito that bites on the infected rabbit becomes colonized and transfers it to another mammal. Pretty simple, right? But this new component here is the flower nectar, and the flower, that's specifically a squash flower, in case you're curious. So the infected rabbit bites, gets bitten, and then intermittent feeding allows deposition of the bacteria in this non-modal source. So now, other mosquitoes, when transiently feeding, will feed on the nectar, become colonized with Francisella tularensis, and carry it further. Additionally, this gives males a role in disease transmission, which they were thought to have no role at all. This gives males a role that, when feeding on flower nectar, they can transfer it to another flower, which is fascinating and has direct implications to what we do today. So why are we hypothesizing this? Francisella tularensis has these characteristics that suggest its ability to survive in flower nectar. These include microaerobiosis and survival in such environments. So being a facultative anaerobe, Francisella tularensis is able to respirate and produce energy in environments with low oxygen. In addition, it produces catalase, which targets hydrogen peroxide, a typical defensive agent of plants and plant nectar. And lastly, it's relatively osmotolerant. So Francisella has an ability to withstand osmotic changes in pressure that could be produced by flower nectar in their sugars. So back in the old days, we used to treat meats with salts, right, and sugar. So that induces an osmotic pressure which disrupts bacteria, and hopefully, we don't have contaminated meat in a sense. But Francisella tularensis can avoid this change in osmotic pressure, which suggests that it could possibly survive in flower nectar. Now, in Thielus et al.'s experiment, we've already showed that 80 species have been linked to tularemia in the Eastern Europe, and this has implications for disease control today. So it's kind of funny now I said that males may have a role, because the way we treat, we target dengue fever or Zika virus or things like that are carried by arthropod-borne vectors, is we irradiate males. So we rear up a bunch of adult males, we irradiate them to make them sterile in a sense, and then release them massively over the environment to hopefully mate with females and produce unviable offspring, right? So that's good if males didn't play a role in mosquito-to-mosquito transmission, but this study could suggest that they do. And in a sense, it was primarily females, so this would be okay because males don't bite. But if the males transfer it from flower nectar to flower nectar, then there could be an incidence in still spreading the disease further. So this doesn't work if Francisella tularensis cannot survive in flower nectar, right? So that's what we want to first test. Does it have this ability to survive in flower nectar? So we chose squash flower, and squash flower is an imperfect flower, which has both male and female flowers separated from one another. And we use these flowers because in the literature they state they have the most copious amount of nectar production, which you're going to find that funny here in a second. But we collect the nectar from the base of the nectary, the female flower, because they produce more nectar than the males. And here's a video demonstrating such. Capillary tube inserted at the base. I use capillary action to suck up the nectar. And you'll see the copious amounts of nectar when I pull it out. That's about 10 microliters. So it's still an arduous process over time. So we take this nectar after about a year of collecting, and we run an experimental setup that allows us to test it against a positive and negative control. So our positive control here is tryptic soy broth with cysteine, and this is a growth medium used for Francisella tularensis that we use for cultures upstairs in the lab. And Francisella tularensis can replicate, grow, and divide in this medium. And then water. So water is a medium that Francisella does not survive well in at all. It actually dies rather readily, if you will. So we take our Francisella tularensis, the live vaccine strain, so we don't kill everybody in West Liberty. We take the live vaccine strain and we inoculate each of these solvents with a 1:10 ratio of such, and then leave those sit on the bench side at room temperature over time to mimic the natural environment and analyze survival over a 7-day period. And here's the graph. So we analyze the survival over a 7-day period, located below, and this is colony forming units. So this is bacterial presence on a plate, which is then multiplied by a dilution factor to get an overall estimation of what is present in the tube. And as you can see, these groupings right here are nectar and TSBC, and they're neck and neck. There's no significant difference between the two, showing that nectar can provide growth as well as TSBC in a sense. And we do have a significant difference between those and water, suggesting that yes, Francisella tularensis can survive with flower nectar. Now that's good. The next step we want to do is we want to have a flower nectar surrogate. So we want to find, yeah.
So Austin, you say that they don't survive in water.
Yeah.
I think that they look pretty, is that out, the time point is days?
Yeah.
It looks pretty impressive that you still have quite a bit alive in water on day 7.
Right. So this experiment has been taken all the way to 21 days.
Okay.
And then that's when we saw complete death of water.
So they don't survive for long periods of time.
Right.
Okay.
So we did this over.
Go ahead.
Okay. We did this over 7 days to mimic like that of a flower, if you will. So how long does a flower last until it's fertilized and then fruit becomes bearing? So it doesn't last very long. So 7 days was an approximate time span that we could use. So,
I'm bring out my college's disclaimer.
okay.
Yeah. Did you by any chance, like the water that you raised it in, did you have any, what was the oxygen content of the water? Was it just like open to the air? Or in a container?
So these were shut conical epidural tubes.
So you said it's a
It's
an
a fact
anaerobic.
tube.
Does it grow better in the absence of oxygen, or
No. So it could.
it can grow in the absence of oxygen, but it
Right.
still kind of sucks?
Right.
Okay.
Right. It's not this like us implementation.
All right. We're good.
So why did you choose that environment rather than putting a light cover on it, a light cap on it that could exchange oxygen?
Because the flower nectar doesn't allow for like permeation of free oxygen. So if the bacteria is embedded in such. Any other questions? So our next goal is, because the copious amounts of nectar is just not enough for us, can we find a nectar solution surrogate that can allow for Francisella survival as we do laboratory testing? And preliminary testing went in before this, and groups before me like Andrea Renshaw and Jenna Wright and Dennis Gooch, they all tested the different concentrations of sucrose that are present in physiological conditions, and that's anywhere from 10% to 80%. And for us, 30% showed a better representation of the flower for survival analysis. And it's not the best, right? We have a significant difference between TSBC and sucrose. So there could be other components rather than just simple sugars like disaccharides that provide a vital role for nectar or for survival in nectar. And additionally, though, we still have a 1.5 log to 2 log significant growth over water, making this usable for further testing. So now that we figured out that we can have a laboratory surrogate to mimic these experimental results, we want to see if these mosquitoes can become colonized after